Okay, guys, so let's go into, now we're going to do a little bit of protein synthesis. Woo! DNA and DNA replication, sorry. So right down here on the left, what you're seeing here is DNA replication in this lower picture. And the reason we know that is if you look here, you see it's splitting the whole thing, and there's a DNA helicase, and what's happening is it's forming a complementary strand on each side. So when you see this occurring like this means it is protein synthesis. I mean DNA replication. DNA replication right here on the lower left. Now, up here in the upper right, if you see these little guys, these are tRNAs. And if you ever see the word tRNA, you need to know we're talking about protein synthesis. You see the words mRNA, tRNA, transcription or translation, all those things scream protein synthesis and ribosomes. Okay, you see all those things? Right away, I want you thinking protein synthesis. All right, so right now, we're going to click and drag. Oh, wait, I don't. That's you guys. Click and drag. Can they not do it? Can I do it?
No, you have to do that.
It won't let me.
You do it with them.
It won't let me.
Okay. Well, okay, no, you can't. I didn't make it so that you can.
Oh.
All right.
Well, we can talk about it.
No, that's fine. So which one, the DNA or the RNA, is single-stranded? I'm sorry, I thought it would be able to let you do that. If you click on it, go into the live version of it.
I'm in a presentation, the Pear deck.
Which one's single-stranded, guys? The RNA or the DNA?
You can put it in chat or anybody. Which one's single-stranded? Take a guess. RNA! Yes, Evan. RNA is single-stranded. Stranded. Remember, because it has a twisted heel. Now, which one has the ribose sugar? Second one right here. Who has ribose sugar? RNA. Yes, Rami. RNA. Think of it because it has RNA. Ribose sugar. RNA. Which one has deoxyribose sugar? DNA. Yes. Now, our zoomies are really coming on strong, but Evan beat you to it, Cora. Double helix. DNA. Okay. Which one has uracil? I switched it up on you so that way you didn't just start writing it. No, RNA is uracil. Yes, Rami. RNA is uracil. Use. T-thymine is on DNA. And I'm sorry this couldn't let us, you know why? Because when we're in the Pear deck, it wouldn't let you manipulate it. Yeah. That's okay. We can talk about it. Good job. So go ahead. Right now, drag red dots to all the characteristics of RNA and blue dots, all the characteristics of DNA. I'll give it a second before I show you what people will put. We'll find out. I'll give it a second. Yeah, it's little things like that when we do these that we always forget. And that's fine. We work through it. Show responses. Let me see. Oh, all right, so I got a blue on, I got a red dot on single-stranded. Ribose is another single, uracil. All right, I got some blue dots on deoxyribose. That's a DNA. Good. Thymine's only in DNA. So get that red dot right. Double. Oh, there's only one dot for each kid. Oh. It's okay because we have multiple ones for different people.
Well, I mean, you're getting them right.
Yeah.
So single-stranded, uracil, and
Ribose sugar. Get those blues away.
the blues should not be on the right.
And this red should not be here.
Right. We got those wrong. So single-stranded, uracil, and ribose should all be red. Deoxyribose, thymine, and double helix should all be blue.
All right.
All right. So thanks for sharing the work on that, guys. That's well done. Nice job.
All right. DNA replication occurs in which organelle? Which organelle? When you're replicating DNA. Think about this. Where is DNA found? And if I said DNA can't leave the nucleus, when you're making copies of DNA, where does it have to happen? Yeah, it was so like, you're like, it can't be that easy, but it is. I get it. Like, yeah. So I get it. So, all right, nucleus, excellent. DNA replication happens in the nucleus. Now, I get it. Some of you, like I was just talking with Evan, he's like, and some of you guys are like, is it that simple? Yes, because DNA cannot leave the nucleus, so DNA replication has to happen in the nucleus. And I just need you to understand there's a differences that are happening right now. And that's the key point to here. So DNA's replication occurs during what cell's life cycle? Ooh, we've been putting this in your head left and right. Anybody remember? Take your chance. Choose an option. Anybody choose an option. So 9 of you are responding. There is 10 people alive. I have the people in class are more alive. Ariel, tell me, which phase do you think it is? No, I appreciate you taking a guess, not M phase, but it is S phase. S phase. S is synthesis, and that's what S stands for. Synthesis means to make. So it's making a copy of the DNA. S phase. And I guarantee that'll probably be a question on the test. If Ms. Murdoch has been saying this left and right, and I've been saying this left and right, there'll be something about this. Well, I'm just guessing. I'm just a man of leisure, but I'm guessing. All right, so unfortunately, we cannot drag these down.
So on the bottom, there is a string of DNA nucleotides that are together. The D's are,
Oh.
Okay, so the DNA is on the bottom. So what you have to do is figure out what complementary bases are going to go with A, C, T, G, C, T, G. Because those are the bases. That's the sequence of bases. The bases that would complement that in the right order. Remember, A always goes with T, C always goes with G. So when you do your lab, you're going to be able to click and drag those pieces over to the right spots. But here, what I'm asking you to do is figure out which sequence of bases goes with that bottom string, A, C, T, G, C, T, G. So now let's take a look at what you got.
Yes, TGAC. Excellent. And why is it B? Because we're talking about DNA. And if you see U's, it cannot be DNA. That.
All right, so we're doing DNA replication here. We're not doing protein synthesis yet. We're reviewing DNA replication. That's the other reason why the DNA omits. So when you're putting complementary DNA bases there, you're going to include the T, the thymine. A would not be right because A would be RNA nucleotides because of that uracil, that U at the beginning. Apple under the tree, thank you, Becky. Beautiful job. All right, let's go to the next slide.
All right. In the diagram you just completed, which enzyme does the work of matching DNA nucleotides? Do you remember this one? Do you remember this one? Which one matches up the free-floating nucleotides? Remember I called it like the grandmother, like, listen, adenine, I got a great thymine for you. I'm going to match you up. I'm going to be great. Huh? Which one do you think of these four do you think is matching it? Huh? You don't remember? All right, it's all right. Just, huh? Take a chance. If it's matching to make DNA, what do you think it is?
They're all enzymes, right?
Huh?
Because they all end in AS.
No. Oh, no. DNA polymerase. And the way you know that is because RNA polymerase will make an RNA strand. DNA polymerase is in charge of making DNA, since it has the name in it. Helicase will unzip regardless, but if you see DNA polymerase, that's the DNA matchmaker. And RNA would be the RNA matchmaker. So it's so easy, it's hard. like you're like, I know I can't be right, but you are. So it's DNA polymerase. Let me see what we got here. DNA polymerase. Yes, you remember her, Pedro. What a wonderful woman DNA polymerase is. She finds you a fine thymine. Listen, I got a guanine that goes to church for you, cytosine. It's a wholesome guanine. You know, you go to church, it takes care of its community. Great guanine. So good. DNA polymerase makes DNA. What do you think RNA polymerase does? Somebody tell me. What do you think RNA polymerase does? If DNA polymerase makes DNA, what do you think RNA polymerase makes? RNA. Yes. RNA polymerase makes RNA. Thank you, Pedro. Thank you, Evan. Yes. Yes, yes. Does the same for RNA. Yes, Banky. The Australians coming in strong. Strong. There you go. Like a kangaroo boxing. I think that's what they do. Don't kangaroos box? I've seen videos.
They fight with each other, I guess.
Yeah. Oh, Banky's South African. Sorry, not Australian. My bad, Banky. I have Australians too. Sorry. I just had an Australian leave my other class, go back to Australia this year. It was really sad. He was a good kid. What is the purpose of DNA replication? Choose a response. What's the purpose? What's the purpose of DNA replication? Come on, my little put-em-theres. Choose a response. 6 of you are responding. What are the rest of you doing? Out to lunch? Say hello. Hello there. In the computer. 12. All right, let's see your response. To make a copy of the cell's DNA in preparation for mitosis. I knew it. You're all smart. You're so smart. You're so smart. So smart. Evan's like, oh my God, this guy's killing me. Poor Evan Matos is here next to me going, please just stop. Please just stop. He judges me. He judges me a lot. Gibson's like, I'm just going to face the computer, not draw any attention to me, and nobody's going to know anything. All right. So first step of protein synthesis, we talked about this in our video premiere, is transcription. During transcription, a small section of DNA is unzipped, and mRNA transcript is made using DNA as a guide. All right, so give me 1 second. I got to share this slide. I got to stop and share a new slide. Oh my gosh, how am I doing that? Let me go up to here, share content. Stop. Share application screen. Chrome tab. DNA mutation simulation. So, what we're going to talk about, click a new to perform. We're going to do transcription first. So what happens? DNA splits. And notice, only one side is made. How do I pause it? How do I pause it? Pause. Stop. Okay, so DNA splits. Thank God. Now, what you noticed is it started pairing up. And we have our G with C, C with G, T with A, C with G, T with A, C with G, G with C. Then we have this A. But when we talk about proteins, it changes to a U. So now we have our uracils. Yep, when you thought you understood everything before, in protein synthesis, when we get to transcription, when you have usually a T, it now needs to be a U in place. So you notice here we have T, it goes to A. That's right. A, and you're like, oh, I know A goes with T. No, if we're talking about mRNA, the A goes with a U, and it now forms uracil. So what this is making is just a code of, and we're just transcribing.
Both sides being copied. You don't have two strands being made. You only have a little bit of the DNA unzipped, and you get enough messenger RNA nucleotides to make that short strand. Now it's going to, and then the next step is it leaves, it goes to the ribosome for the second step.
Should I just show that now before I go into the other one?
No, I would just go on because it's going to ask questions about this first.
Okay.
Good. Follow the order.
Okay. I didn't know. I heard you stop, and I was like, I'll just leave it there and then I come back to it.
Right.
Yeah. I didn't know if you, I thought you were getting ahead of it, and I was like, did you want me to? All right, there we go. So, transcription. Okay, good. I didn't know. She got excited, and I was like, she's saying translation. Do I want to go into that? Oh, my God, no. Okay, I got ahead of myself. Sorry. All right, so transcription, first step. You got to scribe out the code. You got to transcribe the first step protein synthesis. And it occurs in which organelle? All right, where does this occur? Think about it. You got to go to the DNA and get a portion, no problem here, a portion of the DNA. Where is this going to happen? Yes, in the nucleus. Did somebody say that? Somebody say that online too? In the nucleus. First step, transcription, happens in the nucleus because the DNA can't leave. Remember, it's sort of like getting one book. You don't go just get a book from somewhere. You have to go to the library. You have to go to the nucleus to get that one book. You have to go to the recipe book to get the one recipe. All right, good, 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 good. Oh, show response, nucleus. Sorry, I wanted people to just say in person before I even showed it. Yes, nucleus is where this occurs. Very good, very good, very good. All right, transcription. Which of these is the correct order for this? Ariel, let's go back to the page and stop trying to fall asleep. Let's go. Which is the correct order for this? Now remember, we're making RNA. So for making RNA, what letter has to replace the T, Evan? I knew you knew it. There you go, brother. Good job. Good job. I'm just asking you in general, just because I can't. I like you in person. I think you do well. Excellent, excellent, excellent. All right, let me show responses. Let's see what we got. Yes, UGACGAC. If we're talking about mRNA, we need the U's. Very good. Very good. In the diagram you just completed, which enzyme does the work of matching RNA nucleotides? You know this one now, don't you? Tell me. Let's see what we got. Let's see, we got 8 responses. There's 9 people because Evan just told me in person. 9 people are with me here. 8 people online. Come on, my Zoomies. Get up. I know it is. It's Rami sitting at home being like, oh, Mr. Bouchard, I don't want to answer your questions. Banky's too busy doing laps in the swimming pool. Pedro's too busy playing his viola, I believe. Or violin. Shoot. I know it's one of those. Oh, I knew it was a V. I knew it began with a V. He's like, I am done with you, large man. All right, let me show the responses. RNA polymerase. Yes, RNA polymerase is the matchmaker for the RNA. It goes, listen, adenine, I got the perfect uracil for you. You guys are going to make cute babies. You're going to be the best. You're going to make the best proteins if I match you up correctly. Oh, my gosh, I can see it now. You're going to have this white picket protein. It's going to be, you're going to have 2 cats and a dog. You're going to have the best transcription of your life. No, rough crowd. All right, so step 2, during translation, the mRNA transcripts moves out of the nucleus into the cytoplasm and attaches to a ribosome. This is where we've taken now, you've made a copy of that one little part. That one little part will leave the nucleus and go to the ribosome. Because the ribosome, and then tRNAs are sort of like tow trucks. They're going to bring in certain amino acids and meet in the ribosome. And the ribosomes, then they meet up and the amino acids dropped off. The next amino acid will come in and sort of sew in and connect. So tRNA is sort of like a tow truck. It's bringing in the amino acids that match up. And only certain tRNAs will bring in certain amino acids. So you can't just go out there. It's like, hey, amino acid. No, you're not my amino acid. No, you're not the one I like. I like guanine over there. I like phenylalanine over there, so I bring you in. So a certain tRNA will only bring in a certain amino acid. But, and I got a big one, when those amino acids come in, they form something called a polypeptide chain. And why is it called polypeptide chain, you ask, Pedro? I knew you were going to ask the question. That's why I'm saying this, Pedro. It's called a polypeptide chain because they form peptide bonds between amino acids. And that's how they bond together to then form the protein. So let me go back. We're going to show you again. I'm going to stop here for a second. Not stop, but just stop sharing. We're going to go back. And where's my application screen? I'm going to go back now and show you my Chrome tab. DNA simulation. Let's go back. I was playing with this yesterday with Ms. Murdoch, and I sent her an email. I go, oh my God, this is amazing. She goes, I know, I found it. It's great. All right, so translate. So, boom. No, stop. Okay. So what's happened here? This was a tRNA. It brought in the phenylalanine, I think that's what that is. And it then this next tRNA brought in proline. Notice they didn't bond it together. They form a peptide bond between them. Oh, look at that. Another one. Another one bites the dust. Look what's happening now. These are starting to more and more of them, and it's forming a chain of amino acids. I love this diagram, by the way, Ms. Murdoch. I was geeking out with this diagram when Ms. Murdoch sent it to me. Ms. Murdoch sent it to me, and I sent her a message going, this is amazing. She goes, I know, it's the greatest thing. Oh, no, I like them as a, oh, no, I like this too. I like them both accompanying each other.
So the green things are trans RNAs, right? And then the long red line that down there is the messenger RNA. So two types of RNA, and they are coming together in the ribosome, which is the brown, big brown thing behind them. That's a ribosome, it's an organelle. And the ribosome is a place that allows those transfer RNAs to come down one at a time and be guided by the base sequence on the RNA to slowly put a protein together. So the actual point of all of this is that string of beads that's being formed up there that's wiggling around. That string of beads is a string of amino acids. That's going to become the protein that you might find in your hair, like keratin, or the protein that makes your eye pigments, makes your eyes brown and blue, or the protein that makes up your muscles. That's how our cells make protein. They put it together one little bead at a time using the messenger RNA transcript, which ultimately came from your DNA. So there it is. That's the point of having the DNA information, is to direct making of proteins in a specific way. You can have all those thousands and thousands of proteins in you that build your body and also make the enzymes that make the reaction work and do all kinds of important things.
So now we have this chain of amino acids called a polypeptide chain because those bonds right here, these little connector points, those are representing peptide bonds. And do you want me to show the protein? Don't do it yet. I was going to show them how it turns to a protein.
We'll do that.
Okay. She gets me all excited. She's like, stop it. I'm like that dog sometimes that gets really excited to do things. When I saw this, I was geeked. I do. I am a, what is it? There we go. Sorry, I had to click that. Just get very excited about these cool things when we find something new in Ms. Murdoch. I don't know. How'd you find this? Did you just find it? Oh, man, she's
My AP Bio co-teacher is, she finds all these amazing things. I don't know where she finds them.
That's amazing.
I use it for AP Bio and
So
I stole it. I won't take it back.
So here's just another picture, and what happens is, notice there's certain ways these have to meet up, and they bring out certain. So this right here is a tRNA. It's 3 bases, and it has attached to it a amino acid, and it's going to see which one would this go with. This guy would go right over here to this.
Yeah. So this is going to be your lab. You're actually going to do this yourself. We're not going to be able to do that.
So we just learned that blank DNA nucleotides codes for a single amino acid. Thinking back to that picture, look back here real quick. I'll go back a second. How many code for an amino acid. Look at that. How many of these code for amino acids? That's what it's asking. Show my responses. How many do you think, Evan? How many do you think? How many code for one amino acid? Yeah, buddy. Yeah. Miss Murdoch, when we do credit, can you just give him credit because he's been participating with me? Sorry, I just want to make sure you got the credit. 3. So if you look good, 3 amino acids code for that. You got to know it's called a codon on the mRNA side, and the tRNA side is called an anticodon. We'll get more into that, but codon is 3 on the mRNA side, and the anticodon has another 3. I think Pedro was enjoying the simulations we had. He's like, this is funny to watch. All right. So what is the correct order of amino acids that you translated in the previous slide? Ooh, I don't know if I can show response. I'm not going to ask you because this one was kind of hard because you wouldn't remember it. So there would be a glutamine. There after glutamine, this guy would come in. It'd be a glutamine, then it'd be a leucine. Sorry, so that's how you figure out the order. You bring them in one at a time. And then there's some, well, no, I won't get into it. Sorry. Translation occurs in which organelle? Which organelle does translation occur in? So transcription occurred in the nucleus. Remember, we went into the nucleus. We got a copy of a portion of the DNA. That portion, I will give you a clue, leaves the nucleus and goes somewhere. Where? Brother, Gibson, you get a virtual hug. You get a virtual hug. Sorry, I don't mean to leave you out. Yes, Gibson. The ribosomes. Excellent, excellent. Let's see what our responses are. Ribosomes, good. Be careful. Lysosomes looks like the word ribosomes, but it's different. Ribosomes, right? And they're very different functions. Remember, lysosomes contain digestive enzymes for breaking stuff down. No. Ribosomes are used to build stuff up. All right? No. Different functions completely. Ribosomes build. All right. Which of the following would not be a product of a cell performing transcription and translation? Would not be a product. Choose an option. Choose one person. One person. I know Pedro is playing the cello right now because he's trying to diversify his stringed instruments, so he's not even participating. I know how it goes. Rami's kicking a soccer ball. Evan's sitting here going, please, let's end this now. Please stop calling on me. Let's see what we got here. No, I said you're trying to diversify your stringed instruments. So you're now just doing the cello for fun. All right, you are glucose, insulin, enzyme, hemoglobin. One of those would not be made. Which one? Yeah, sorry. Which one is not? Enzymes are proteins, guys. Enzymes are not proteins. I mean, enzymes are proteins. Well, enzymes are proteins. Insulin is a type of protein. Hemoglobin is a type of protein. What is glucose? What is glucose? Sugar. All right, guys. So glucose. Thank you, Pedro. Thank you. Look at that. He can learn a new instrument and answer a question at the same time. Brilliant. Maybe you should do the recorder. Okay, I've talked enough. Let's take a break for a second.